Inviting me to come and speak. Hope you feel better soon, Sarah. So we've heard throughout the afternoon that modern humans overlapped in time and space with multiple hominin lineages. And one topic that has been of enduring interest is, was there any admixture that happened between modern humans and these other groups of humans? And for a long time, the answer was, well, maybe yes and maybe no, and people debated it pretty vociferously. And that was largely because the data to answer that question didn't exist. And it wasn't until more recently that Svante Pääbo's group in Leipzig developed and pioneered tools for studying ancient DNA. And they produced the first Neanderthal genome sequence. And we finally had the tools to be able to say definitively whether admixture occurred or not. And indeed, as Shriram talked about earlier, all non-Africans derive about 2% of their ancestry from Neanderthals. And what was even more interesting is a few years later, this same group published another paper where they sequenced ancient DNA isolated from a small fragment of a pinky bone, thinking that it was perhaps Neanderthal or maybe modern human. And it turned out to be this entire new branch of humanity that we now call the Denisovans. And this is, in fact, the first species to be entirely described by DNA. And so ancient DNA has transformed our understanding of human history over the past decade. And we've learned many things, like what the distribution of Neanderthal ancestry is in populations across the world. And again, we saw this this morning, but we can see that on average, individuals outside of Africa can trace about 2% of their genomes back to Neanderthal ancestors. And strikingly, we find a very different picture for Denisovan ancestry. So here, we really only find Denisovan ancestry in parts of the world, down here. Oops, that's not working, so I'll just talk. In populations of Melanesian and Australian Aboriginal origin. So we have a very different geographic pattern of surviving ancestry. And that's great. We can describe global ancestry proportions, but studying ancient DNA is still hard. So my interest in this area can be traced back to a few years ago, where we had this idea that, well, if modern individuals interbred with Neanderthals and Denisovans, then maybe we don't have to excavate ancient DNA directly from fossils, but we could indirectly isolate Neanderthal and Denisovan sequences from the genomes of modern humans. And so we call this molecular excavations. And I borrowed this slide from a CARTA meeting a few years ago, which I think is a really beautiful representation of this idea of molecular excavations. And so literally what we're trying to do is develop computational or statistical models and walk along somebody's genome and pull out the bits that were inherited from Neanderthals or Denisovans. And molecular excavations are really powerful because they enable us to identify the specific DNA sequences that were inherited from Neanderthal or Denisovan ancestors. So it's one thing to say something about a proportion, but when you can actually identify the sequences, you can do a lot of interesting things with it. So you can test evolutionary hypotheses, and you can even start thinking about, well, what's the influence of Neanderthal and Denisovan sequences on traits and diseases in present-day populations? And I'll try to touch on all of this in my talk. So we've discovered methods to identify Neanderthal and Denisovan sequences, and we've applied them to geographically diverse populations. We've largely looked at around 2,500 genomes that are part of a publicly available project called the 1,000 Genomes Project. But we've also worked with colleagues in some cases to sample populations from particular regions of the world. For instance, Melanesia, where we expect Denisovan ancestry to be the highest. And so how much of the Neanderthal and Denisovan genome persists in modern individuals? So if we just represent the Neanderthal genome as this circle, when we look across all 2,500 people, we actually recover about 41% of the Neanderthal genome. And that's pretty striking, right? That we're not actually sequencing a Neanderthal, but we're stringing together these bits and pieces that survive in modern individuals. And by doing that, we can find almost half of the Neanderthal genome. And that might seem surprising, especially in light that each of us only carries a little bit of Neanderthal ancestry. But the reason this works is that the 2% of Neanderthal sequence that I have might be a little bit different than the 2% that you have. And when we look collectively across large numbers of individuals, we can recover a substantial amount of the Neanderthal genome. And on an individual basis, non-African individuals have about 55 million base pairs of Neanderthal sequence per individual. And this is pretty similar across populations, so East Asians, South Asians, Europeans, and American individuals. There's a little variation, but it's fairly consistent. And incidentally, if you get your 23andMe report and they tell you you either have the most Neanderthal ancestry or the least Neanderthal ancestry, what it's really saying is that if you have the least amount, you have about 40 megabases of sequence, and if you have the most, you have 60 megabases of sequence, and whether that's interesting or not, that's entirely up to you. So we can do the same thing for Denisovan sequences. Again, represent the Denisovan genome as this circle. And here, we don't do quite as well, but we still recover 10% of the genome, which is a substantial amount. And the reason we don't recover quite as much is that Denisovan ancestry is largely confined to Melanesian populations. So in fact, Melanesians have about 40 megabases of Denisovan sequence per individual, and you find very little Denisovan sequence in other populations. And in fact, this 10% number is actually pretty good because we only have a sample size of 35 individuals compared to the 2,500 individuals that we're looking for Neanderthal sequence in. So in fact, there's a lot more of the Denisovan genome to be found. So that's interesting. We can identify intragress sequence. But really, what we're interested in is understanding whether admixture was just an interesting side note to human history, or was it something more significant? And in